The female orgasm is frequently misunderstood, often simplified as a singular event, yet it is one of the most complex biological phenomena in human physiology. It is not merely a physical reflex, but a sophisticated interplay between psychology, neurology, and vascular biology. To understand the female orgasm, we must first look beyond the genitals and observe the body's true control center, the brain. While the physical sensation may be localized, the experience is orchestrated by the mind. The limbic system, the area of the brain responsible for emotion and motivation, works in tandem with the prefrontal cortex. For an orgasm to occur, the brain must often disengage from daily stressors, a process known as transient hypofrontality. This quieting of the analytical mind allows the sensory information to take precedence. However, this biological pathway is unique to every woman. The threshold for arousal, the time required to reach peak excitation, and the type of stimulation needed are not standardized. Factors such as mental health, hormonal balance, and even the level of trust in a partner play a critical role in the biological availability of pleasure. The brain acts as the gatekeeper. If the psychological environment is not conducive, the biological cascade may never begin. For decades, medical science minimized the importance of the clitoris, often describing it only as the small, visible glands. Modern 3D imaging has revolutionized this understanding. We now know that the clitoris is a vast, complex organ, the majority of which lies hidden beneath the surface of the skin. It is analogous to an iceberg. What is visible is only the tip. The external glans is the point of highest nerve density, containing upwards of 8,000 nerve endings, double that of the male penis. However, beneath the surface lies the clitoral body, which divides into two crura, or legs, that stretch backward toward the sight bones. Flanking the vaginal opening are the vestibular bulbs. These are crescent-shaped structures composed of erectile tissue. This entire complex is homologous to the male penis, meaning they develop from the same embryonic tissue. When a woman becomes aroused, the role of this internal structure becomes clear. The legs and bulbs fill with blood, a process called vasocongestion. As they swell, they hug the vaginal canal and the urethra, tightening the internal space and creating a cradle of high-pressure sensation. This anatomical reality explains why vaginal stimulation is often actually internal clitoral stimulation. The vaginal canal itself is a dynamic organ. While the outer 1/3 of the vagina is rich in nerve endings, the inner 2/3 are less sensitive to light touch, but highly responsive to pressure and stretch. This leads us to the controversial but anatomically significant area known as the G-spot, or the urethral sponge. Located on the anterior or front wall of the vagina, this area is not a distinct button, but a zone of tissue that is part of the clitoral network and the periurethral glands. Stimulation here triggers a different neural pathway than external touch. While clitoral stimulation travels primarily via the pudendal nerve, internal stimulation engages the pelvic and hypogastric nerves, and even the vagus nerve, which bypasses the spinal cord and connects directly to the brain. Further deep is the cervix, the gateway to the uterus. During high arousal, the uterus undergoes the tenting effect. The muscles of the pelvic floor lift the uterus up into the abdominal cavity, pulling the cervix away from the vaginal floor to create a longer canal. Stimulation of the cervix can trigger deep, visceral orgasms for some women, owing to its unique connection to the vagus nerve, proving that the pathways to pleasure are multiple and redundant. The transmission of pleasure is an electrical event. The body is mapped with erogenous zones, areas of the skin with heightened sensitivity. While the genitals are the primary focus, the neck, ears, breasts, and inner thighs are covered in receptors that feed into the same arousal loop. When a nipple is stimulated, for example, it sends a signal via the intercostal nerves to the spinal cord. These signals race upward to the genital sensory cortex in the brain. Interestingly, MRI scans have shown that the area of the brain that lights up during nipple stimulation is the same area that activates during genital stimulation. This neural traffic is intense. Millions of bits of information are processed per second. The sympathetic nervous system, usually responsible for the fight or flight response, begins to accelerate heart rate and breathing, while the parasympathetic system, responsible for rest and digest, mediates the blood flow necessary for arousal. It is a delicate balancing act of the autonomic nervous system. As the nervous system fires, the vascular system responds. The heart beats faster, pumping oxygen-rich blood to the pelvis. This increase in localized blood pressure causes a phenomenon known as transudation. The vaginal walls do not have glands to produce liquid directly. Instead, the pressure forces plasma from the blood vessels to filter through the vaginal epithelium. This creates a slick, clear layer of lubrication on the surface of the vaginal walls. Essentially, the vagina sweats in response to arousal. Simultaneously, the Bartholin's glands, located at the vaginal entrance, secrete a mucus to aid in reducing friction. But there is another fluid phenomenon that has sparked scientific debate for years: female ejaculation. Surrounding the urethra are the Skene's glands, often referred to as the female prostate. In some women, these glands are highly active. During intense stimulation of the G-spot region, these glands can fill with fluid. At the moment of orgasm, they may expel this fluid through the urethra. Chemical analysis has proven that this fluid is distinct from urine. It contains high concentrations of prostatic acid phosphatase, PSA, urea, and creatinine. It is a functional secretion unique to the moment of sexual climax. The orgasm itself is the explosive release of the accumulated neuromuscular tension. As the arousal phase hits its peak, often called the plateau, the body is primed. The muscles of the pelvis, specifically the ischiocavernosus and bulbospongiosus muscles, are taut with energy. When the threshold is crossed, the brain triggers a reflex cascade. The orgasm manifests as a series of rhythmic, involuntary contractions in the vagina, uterus, and anal sphincter. These contractions typically occur at intervals of 0.8 seconds, gradually slowing in frequency and intensity. Neurologically, this is a storm. The brain releases a powerful cocktail of neurochemicals. Dopamine creates intense pleasure and craving. Oxytocin, the bonding hormone, fosters a sense of emotional connection and trust. And endorphins act as natural painkillers and sedatives. For a brief moment, the brain's decision-making centers go offline, creating a state of pure sensory experience. Following the peak, the body enters the resolution phase. The blood that had pooled in the genitals begins to disperse, returning to the rest of the circulation. The deep red and purple hues of the vulva fade back to pink. The heart rate and breathing slow to baseline levels. Here lies a significant biological difference between males and females. most males enter a refractory period, a time during which physiological recovery prevents further orgasm. Women, however, largely lack this refractory period. If stimulation continues and the arousal levels remain high, many women are capable of experiencing multiple, sequential orgasms before the resolution phase is fully complete. However, the ability to reach this state is fragile. External factors such as stress, fatigue, antidepressants, specifically SSRIs, and past trauma can disrupt the neural pathways. Conditions like vaginismus or pelvic floor dysfunction can turn this pleasure into pain. Conversely, a feeling of safety and emotional intimacy can act as an amplifier, enhancing the biological response